Hello, welcome to part 2 of chapter 19, um, dealing with electrical impulses within the heart. This is Professor Young, and we'll be spending about the next half hour or so working through some of the information. One of the key things that you need to understand is that as we move through this material in chapter 19 and spilling over into a little bit of chapter 20, we need to look at electrical impulses from the view of anatomy. How exactly does the action potentials that are generated impact the various chambers associated with the heart, and what is the result of that impact? And so that's, by the end of this WIMBA session, that's exactly what we're going to be doing. We're going to be tying all of this together, hopefully into one nice, neat little cohesive package that'll have you be able to go out and explain this to anyone that you come across. Because after all, being able to teach material to someone else at your level is, I think, the highest level of understanding that one can have. And so that's our goal and our achievement as we go through this information. Some basic information, most of this of which we have already gone through. We know that cardiac cells have special areas, really gap junctions, called intercalated discs. And within these intercalated discs, we're really talking about an area within cardiac cells that allow muscular tissue, cardiac muscular tissue, to communicate with one another. And there's really three key regions to these intercalated discs. We have the gap junctions themselves, which serve the purpose that really provides the electrical stimulation to coordinate the contraction within both the atrium and the ventricles. While the ventricles provide the majority of the force of the contraction within the heart, we cannot forget that the atrium also does exhibit some contractile force within it. And so gap junctions really provide that communication between cardiac cells above and below where the gap junctions are located. We also have, if we remember, desmosomes that are located within these gap junctions. And again, the desmosomes really connect, linking or side-by-side cardiocytes together. And so that way, during the contraction and relaxation phase, especially within the ventricular walls, these cardiocytes or myocardial cells do not separate during contraction and there again relaxation. And so if you think of two cardio cells or cardiocytes side by side, they're going to be connected with these desmosomes. Now, inside of the desmosomes are these fascia adherens, what I typically refer to as just being adherens. And the role or the job of the adherens basically are to act as these little tiny rubber bands located within the desmosomes. And so that during contraction and relaxation, the desmosomes are stretched and pulled, and the adherens stretch with it. And again, that allows for those desmosomes to really stay intact. Kind of think of the adherens as if you ever look at those big bouncy balloons that have the large rubber band on the end that you can sit there and punch. And as you punch that balloon, the rubber band stretches, which allows the balloon to go out, but it also then pulls it back in. That's the job of the adherens within these intercalated discs. when we talk about intercalated discs allowing for intercellular communication between the cardiocytes, what we're really referring to here is the passing of the action potential. And if you recall, and if you need to, I would suggest go back and review this from your discussion in AMP1 of muscle contractions. But we know that as a action potential is moved down the sarcolemma, the cell membrane of a muscular cell, that sodium basically diffuses very rapidly into the cell, and that then projects, if you would, the action potential down that sarcolemma. We see the same thing as an action potential is passed down an axon. At the areas of the nodes of Envisioning, sodium rushes in, which again propels that action potential down. We see that same type of action here found within the intercalated discs. And again, what this basically allows to happen is for all of the cells to be able to communicate with one another and function as a single cell, so that all of the cardiocytes found within the myocardium of the ventricle can function as one ventricle, and likewise with the right and the left atrium. And so that's key. And that term that we use to describe the ability for those cells to not only generate its own response, but to communicate effectively as one cell is autorhythmicity, which you see that term autorhythmic right there. And so that is key. That is key for the function and the ability of the heart to be able to do what it does. And so again, the cardiac cells, the cardiocytes, are extremely permeable to sodium, as I've already mentioned, and this will lead to the action potentials moving through the intercalated discs, therefore allowing the cardiocytes to communicate with one another, leading to one strong coordinated contraction. Now, you're probably asking yourself, well, how exactly does this action potential get started? If the heart is a self-contained electrical generator for its own impulses, it's not going to take its cue from the nervous system. So in other words, a action potential from the brain is not going to be sent to the heart to tell the heart to pump. The heart needs to generate that action potential, that electrical stimuli, from within itself. It does that through a couple different, or at least it does that through one key structure, and then that action potential is sent through a couple other various structures that we need to look at and be familiar with. First of all, found within, and this is key, found within the right atrium is the SA node, the sinoatrial node. And so that would be right here. And the purpose of that sinoatrial node does just that. It stimulates or initiates the electrical stimulation for that action potential to be sent. It is found in the wall of the right atrium. It is found in the superior area of the right atrium. So it's found at the top of the right atrium. That electrical stimuli is sent from it. Part of that action potential is sent to the left atrium. Part of that is sent to the remainder of the right atrium, so that your atria can contract to make sure that all of the blood that is secured within it flows down and into the ventricles. But part of that action potential, leaving the sinoatrial node, the SA node, also then travels to the SA node, the AV node, the atrioventricular node. And so that's key for us to understand that once the SA node fires, it sends that impulse to the left atrium, it sends it to the right atrium, and it also sends it to the AV node, the atrioventricular node. The SA node is not the end-all and be-all for generating this response. If the SA node fails, or if it wears out, prior to receiving a pacemaker, and all the pacemaker does is the pacemaker acts as the SA node, the AV node can act as a backup. The downside to that is the SA or the AV node is not as efficient and not as effective at producing that impulse on a regular interval. And so it tends to respond slower, which then again would slow down the basic overall operation of the heart, which is not a good thing. And so normally the SA node would fire the action potential and will go to the AV node. And it will also go to the left atrium and the right atrium respectfully. That aside, if we just follow the path of the action potential so that we can get it down into the ventricles, because keep in mind, the overall goal here is to allow the ventricles to contract. That is where the majority of the force comes from within the heart when we talk about the contraction of it. And so that electrical impulse leaving the SA node needs to get its way down to the ventricle. So SA node impulse is sent. It is received at the atrioventricular node. The AV node will then pass that electrical impulse, that action potential, down to the atrioventricular bundle, which splits into the right and the left branches, respectively. The right atrioventricular bundle will go ahead and initiate a contraction within the right ventricle. The left atrioventricular bundle would split and basically coordinate contraction within the left ventricle. Another term for the right and left atrioventricular bundle is the right and left, respectively, the right and left bundle of His. And so very often you'll hear that AV bundle being referred to as the bundle of His, and then whether it's going right or left. But that AV bundle will split into the right and left branches and go to that respected or that specific ventricle, whether it's right or left. From there, once sent down the right and left bundles of His, it will encounter what we refer to as the Purkinje fibers, and it is finally here within the Purkinje fibers where we see the myocardium of the ventricles contract. The Purkinje fibers are cardiac cells. This is key. I want you to understand this. I want you to live this. I want you to breathe this. I want you to do whatever it is that you need to do. Purkinje fibers are cardiac cells. There it is, right there. All right, so please keep that in mind. But they are specialized cardiac cells. Cardiac cells, being muscle cells, we know initiate contraction. Purkinje fibers do not. Purkinje fibers are cardiac cells that carry electrical stimulation. They carry the action potential. As they are carrying that action potential, they stimulate the contraction of the other myocardial cells around them to then go ahead and lead to the contraction of the ventricles. So if we put all of this together, this is what we see. This here is what we are looking at. And this is where I want to draw our attention for at least a few minutes. Here we have the SA node, and it is the job of that SA node again, to send electrical stimulation to the left atrium as well as to the right atrium. And so there's got to be some muscular contraction that goes here as the atria are filling up, pressure increases, and it's going to flow through either the tricuspid or the bicuspid valves. There's a little extra squeeze that happens within the atria. That stimulation comes from the SA node. What we are more so concerned about, though, is the impact that the SA node has on the AV node, which is where that stimulation then goes. From the SA node, or from the AV node, that stimulation is then received by the atrioventricular bundle, which will then branch off into the right bundle of his, down into the Purkinje fibers, and or the left bundle of his, and into the Purkinje fibers there. Once in the Purkinje fibers, the ventricles will contract. So if we think about this, the atria contract together. That is stimulated by the SA node. The ventricles contract together. The SA node sends the action potential to the AV node, down to the atrioventricular bundle, down to the right bundle of his and the left bundle of his, into the Purkinje fibers, which then stimulate the myocardial cells found within the walls of the ventricle to contract. And they need to do that together. As blood is leaving the right ventricle, blood needs to leave the left ventricle. Otherwise, you have a backlog of blood. You have blood that's still sitting in one ventricle, blood leaving another ventricle. You throw off the pressure, and you've got yourself a big old mess when it comes to cardiac contraction, which you don't want to do. Since we are looking at this view of the heart, you should be able to sit here right now as we speak and identify and label the various structures. In fact, I would suggest that you pause this presentation and try to do that now. You should be able to identify the inferior and the superior vena cava. You should be able to identify the aortic arch. You should be able to identify pulmonary arteries, both the right and the left branch. You should be able to identify both the right and left branch of the pulmonary veins. You should be able to identify the right and left atria, the right and left ventricles, the interventricular septum. You should be able to identify which one's the bicuspid, which one's the tricuspid. You should be able to identify the semilunar valves, both the atrial semilunar and the pulmonary semilunar. You should be able to see in this picture very clearly the myocardial layer, the epicardial layer, the endocardial layer. You should be able to see within this view papillary muscles. You should be able to see chordae tendineae, so on and so forth. You should be able to identify those features now as they relate to the conduction system. And so again, I would suggest pausing this and going through and seeing if you can do that. If you can, continue forward with the presentation. If you can't, go back to your notes, go back to the part 1 of chapter 19, go back to that WIMBA session, and go through that again until you know these parts down cold. You need to be having nightmares of these different structures coming into your sleep and attacking you. That's how well you need to understand this. And so again, pause now to go forward and do that. The rest of us are going to continue on. The cardiac cycle, when we look at this complete cycle, we need to start understanding this from the perspective of not only anatomy, but how pressure is differing within these anatomical regions or chambers of the heart. The entire cardiac cycle takes approximately 1 second to complete, from the time the blood enters into the right atrium until the time it leaves through the aortic semilunar valve. takes about 1 second. And so you need to have this understood. You need to have this appreciation for pressure and how that pressure changes as it goes through the heart. And so here are some key terms: atrial pressure, ventricular pressure, arterial pressure, both systole and distally, and isovolumetric. Again, atrial pressure. This would be the pressure in the right and the left atrium. Ventricular pressure, this would be the pressure found within the right and the left ventricles. Arterial pressure, pressure within the arteries, within the aorta, as well as the pulmonary trunk. Systole, this here would be the pressure in the ventricles. And that's key here to understand. We're talking about ventricular pressure here. If I could spell, that would be so much better. We're talking about ventricular pressure here, but it's the high pressure phase of ventricular contraction, whereas diastole would be the low pressure phase of ventricular relaxation. We will see in a little bit that there is a phase or a brief moment in time during this cardiac cycle when there is no change in volume. There is no change in pressure. Basically, the atria and the ventricles are relatively even, so to speak, when it comes to pressure. And so we will look at that when it comes. So the first three are atrial pressure, ventricular pressure, and arterial pressure deal strictly with the pressure found within the various regions or chambers of the heart, whereas systole, diastole, and isovolumetric deals strictly with the pressure the pressure found within the ventricles during both the phase of contraction and the phase of relaxation. Now, let's take a deep breath because we've covered a lot of material so far. I would suggest before you go on with this Wimba session that if you need to go back and restart this from the beginning to go through the flow, that you do that now before we go into anything else with the cardiac cycle, because we're going to build upon exactly what it is that we have looked at. And so if you need to stop and go back to the beginning, I would suggest doing that now. If you feel you have a good grasp and you are brave enough to venture forward to the next slide, then we will do that now. And so you have that option. The cardiac cycle is broken up into really three different phases. We have ventricular filling, which you see before you. We have ventricular systole and ventricular diastole, respectively. Each of them have different events that happened during those periods of time. Each of these parts of the cardiac cycle relate back to the conduction system that we went over earlier. Each of these is going to have a specific impact found within the various chambers of the heart. And so this is why you need to have that grasp of the anatomy with a good grasp of the conduction system so that we can put both of those together to understand this complete picture of the cardiac cycle. So during ventricular filling, what we see happen here is we see low pressure within the ventricles. We see an increase in pressure within the atria. So what this tells us is that at the beginning of this phase that we're calling quote-unquote ventricular filling, At the start of this process, the ventricles are empty, and the inferior and superior vena cava is now funneling blood into the right atria. The pulmonary veins are now funneling blood back into the left atrium. And so the atria are filling. As the right atrium is filling, pressure is increasing. That pressure is applying a force onto the SA node, the sinoatrial node. That pressure being applied to the SA node triggers the action potential to begin. So that's the first thing that you need to make sure you understand. Is that as the atrium is filling, and specifically the right atrium, pressure is being applied to the SA node, which is going to send the spark to trigger the action potential to begin for the conduction system. At the same time, the atrioventricular valves open. The tricuspid valve is going to open in order to allow blood to flow from the right atrium into the right ventricle. The bicuspid valve is going to open to allow blood to flow in from the left atrium down into the left ventricle. So this is happening simultaneously. How does that happen simultaneously? As the SA node, that sinoatrial node, fires. Remember back on the slide that I had for the picture of the conduction system, we know that the SA node is sending a signal from the SA node over to the left atrium. It's also sending that signal throughout the right atrium that's telling both atria at the same time, squeeze. It's a little squeeze to make sure that all of that blood is now going into the ventricles. Towards the end of this phase, the ventricles are filling, hence why we call this ventricular filling. Right. This is considered diastasis. There is no contraction going on. The ventricular muscles are relaxed, and pressure is increasing within the ventricles. By the end of this process, you are in atrial systole. This is all happening together. This is all happening within probably 10 to 15 milliseconds as far as the beginning of this contraction cycle goes. Right, so this is all happening very rapidly. Atrial systole, pressure drops, ventricles are filling, SA node is firing, atria are squeezing, pressure increases within the ventricles, pressure is decreasing within the atria, both the left and the right atriums separately. Right, so that's what's taking place here first. Then we enter into true ventricular systole. Pressure is increasing rapidly within the ventricles. The SA node has fired down to the AV node. The AV node has sent that impulse over to the atrioventricular bundle, down the right bundle of his, down the left bundle of his, around the bottoms or the apex of the ventricles, into the Purkinje fibers. The Purkinje fibers have passed that on into the myocardial of the ventricle wall, and it's forcing the ventricles to contract. That blood is not sitting in those ventricles for very long. It's not sitting in there very long. The semilunar valves are closed. The atrioventricular valves have closed behind this blood leaving the atria. The pressure is increasing. The Purkinje fibers have sent that impulse on. Bam, you've got the contraction going all on at the same time, which is going to force the semilunar valves to open, and blood is ejected from the ventricles. If it's the right ventricle, the blood is going to be ejected into the pulmonary arteries so that the blood can go to the lungs in order to be reoxygenated. If the blood is leaving the left ventricle, it is being ejected and propelled into the aorta and into that aortic arch so that it can go into the brachial plexus, down up into the shoulders to supply the arms. It can go up into the carotid artery so that it can supply blood to the head, and down into the systemic artery so that it can supply blood to the core of the body, mainly the digestive system, as well as the lower legs. The ventricles are only expelling about half of the blood, maybe 3/4 at the greatest amount. Otherwise, you would lose total pressure within the ventricles, and it would be almost like, what's it called, when you siphon out like a fish tank. All right, and you get the pressure and then you get the flow going down. Once you break that siphoning pressure, you have to restart everything. You don't want to break the siphoning pressure going on within the ventricles. And so you always leave a little bit of pressure building within the ventricles. And so only about half of that total blood is going to be ejected. Then we come to ventricular distally. The action potential is done. The blood is left. And so the ventricles are taking a split-second relaxation break. The pressure within the ventricles have dropped below the arterial pressure. The semilunar valves close. And basically they await for the next firing of the SA node to begin the process all over again. Now, what does this look like altogether? Well, this is it. All right, now, before I go any further, let me make it clear that you need to understand this process in your sleep. I would almost suggest that at this point in time, you kind of close your eyes, lean back in the chair, take a couple deep breaths, and begin to envision this process. Get that mental picture of the heart in your mind there. And you can look down into the heart. So it's cut there in half. You're looking at the right atrium. You can clearly see the SA node. You can clearly see the AV node. You can see the superior and inferior vena cava. You can see the tricuspid valve and the chordae tendineae. You see the right ventricle and the little saxophone shape that it makes as it goes up into the pulmonary semilunar valves. You can see those pulmonary arteries kind of going away from the heart, but you kind of lose them there as behind the heart. You kind of lose them as they go to the lungs. But yet, over on the other side, you can see the left atrium, and you can see bits and pieces of the right and the left pulmonary veins as the blood begins to come back into the left atrium. You can see the bicuspid valve or the mitral valve. And again, you're looking at the chordae tendineae and the papillary muscles of the left ventricle. And that reverse saxophone shape as the pathway from the left ventricle leads up into the aortic semilunar valve, leading into the aortic arch. And from there, you kind of lose the blood vessels as they leave the aortic arch. And so coming back over to the other side, you can actually envision, you are looking at and witnessing blood flowing in from the body through the inferior and superior arch, or inferior and superior vena cava. You can see that blood now filling the right atrium. And you see these little lightning bolts begin to leave from the top portion of the right atrium and spread across to the left atrium. These lightning bolts are now firing out of this top portion of the right atrium, and it's spreading throughout the right atrium. That generator of the lightning that you are looking at, that you're envisioning, those bright flashes of light going across your vision field right now, that is the sinoatrial node. And then every now and then you are seeing that there's a lightning bolt, there's a streak of lightning that is coming out of that SA node, and it's reaching the AV node. And as it's reaching the AV node, you're noticing that the right atrium, it's contracting, and that the tricuspid valve is now open, and that blood is now flowing down into the right ventricle. And that as that blood is flowing into the right ventricle, that you are beginning to now notice that lightning bolt that struck the AV valve, that lightning bolt is now zipping its way out of the AV node into the atrioventricular bundle. The tricuspid valve is now closing between the atria and the ventricle. And that lightning bolt is now traveling down the right bundle of his, and it's reaching down into the lower portion of the apex of the ventricle, and it's coming up into those Purkinje fibers. You can follow that bright light going down, up and around into the Purkinje fibers, and you can see that electrical field being passed throughout all of the myocardial cells that are found within that myocardium layer, that thick area of muscle surrounding the ventricles. And that muscle begins to contract, and that blood that is now pooling within the ventricles are being ejected and propelled at a great force out into the pulmonary arteries, the pulmonary arteries reaching the lungs. And you lose that flow of blood, but then all of a sudden you pick it back up as it's entering back into the pulmonary veins, and it's entering into the left atrium. And that blood is then building pressure again. And you notice some more white electrical lights. Lightning bolts coming back over. You don't quite know where they're coming from, but you see that they're coming across and they're going into that left atrium. And you realize that that's the next round of stimulation coming in from the SA node. And it enters down into the bicuspid valve, down into the left ventricle. And again, you see that white lightning streaking down that interventricular septum, down into the left bundle of his, down around that apex of the ventricle, back up into those Purkinje fibers. And again, as a network of almost like a spider web, you see that white light, that electrical impulse, going out through all the myocardial cells, causing the contraction within the left ventricle. And that blood leaving the left ventricle up into the aortic semilunar valve and then going out into the body. You need to be able to, at some point in time, describe that to somebody. Grab your mailman as he's delivering your mail. Grab the newspaper person that's delivering your newspaper 5 o'clock in the morning. Grab your brother, grab your sister, grab your grandma, grab your mom, grab your wife, grab your husband. I don't care who you grab. But grab somebody and be able to take the time to explain it to them just as I did there. If you can do that, I will not in any possible way be able to trick you on your exam. But if you can't take the time to do that, I will trick you. I guarantee it. You need to be able to envision this process. You need to be able to explain this process to somebody else. It's not enough to read your notes and flip the page, because I can guarantee you you're not going to understand it. Talking through it out loud with someone else in the room, you will catch what you know and what you don't know, and you will be able to correct it. Using this as your guide, this image that you have here in front of you, you will notice that there are a series of colors as we conclude up this little session here. You will notice that you've got the blue area right there, and that is going to be representative down here. Wherever you see the color blue, you know that is where contraction is going to happen. Where you see the pink, you know this is where those vessels are going to be relaxing. Where you see the green, you know those atrioventricular valves, the bicuspid and the mitral valves, are going to be open, or where those semilunar valves are going to be open. And where you see yellow, you know that those semilunar valves or those atrioventricular valves will be closed. Make sure you can follow this picture, right? Because basically what we are seeing here is this. We see in this first image that both the right and the left atrium are filling. This is exactly what I've been going over. As that is happening, the impulse again is being sent from the SA node down through both of the atria, and it will also end up at the AV node. Notice that the atria is filling, and at that point in time, both the valves are open. As that opens up, the ventricles begin to fill. As those ventricles are filling, the AV node is firing off to the, that's a horrible color to try to use. You are noticing that the AV node is firing off to the AV bundle, down to the right. You are noticing that at that point in time, this is all happening all at once, the ventricles begin to contract, and as they begin to contract, again, the AV nodes are closed, but we see that the semilunar valves are now open. It is at this point in time, I want you to look at this picture. Isovolumetric pressure. Everything is closed. There is no blood. There is no blood. The muscles relax. The pressures are, for the most part, even. And then what we see happening here is basically late ventricular diastole combined with, sorry about that, basically blurs into, if you would, atrial systole. And you have both the AV valves beginning to open as response to the atrium's filling. And then the SA node firing to the AV node, and the AV node firing to the atrioventricular bundle. You can see how this is very repetitive, right? But you need to take that time to be able to learn it. So, as I wrap up here and try to calm down from my enthusiasm and slight excitement over this very mundane and boring and yet practical series of events that happens within our heart, run around your house, wake up the dog, wake up the neighbors, call the neighbor over have a pot of coffee with some Kahlua or something like that, and I don't know, whatever you need to do, but begin to explain this process. If you're walking on campus, grab a security guard. They're not doing much except standing around looking for bad people. Most of the people on campus aren't bad, so they're bored. Grab them and say, hey, have you ever wondered how your heart beats? Let me explain it to you, and go through that process with them. If all else fails, stand up in the middle of lecture and scream it at the top of your lungs so that we all know that you know it, or so that we all know that you don't know it, one or the other. And on that note, I will leave you alone for a little bit. Again, as always, continue to study and review. If you have any questions, please feel free to email me and or ask me during class. Until then, happy studying.